decided to use candy to represent our models. Each model is labeled for clarity and will be explained in the video.
In a eukaryotic cell, DNA is not completely laid out as one double helix in a chromosome. DNA must be packaged so that a 6-foot strand can fit inside each cell. Histone proteins bind with DNA to form highly condensed structures of chromatin. A complete histone structure is an octamer formed by 2 tetramers of histone subunits. The 4 core subunits of each tetramer are H2A, H2B, H3, and H4. When DNA wraps around a complete histone structure, the combination is called a nucleosome. Nucleosomes are connected by linkage or spacer DNA and form a fiber that is described as beads on a string. H1 is an accessory enzyme in between nucleosomes controlling the orientation of linkage or spacer DNA. These beads on a string line up next to each other, forming a 30 nanometer fiber, which then forms loops. A sequence of loops then orient until it forms a 300 nanometer fiber and the structure of a chromatid.
A chromatid which has been tagged for protein will not appear uniform along the entire structure. As it turns out, chromatin varies in its amount of histone proteins, and these proteins tend to obstruct reading of the DNA. An area of chromatin that is histone dense is called heterochromatin and contains repressed genes. However, euchromatin is less dense with proteins and contains genes that are open for reading. Using a technique called GM-cis staining, which tags for protein, G-bands form on metaphase chromatin, showing the areas of high and low protein seen here.
Chromatin and histones are heavily studied in epigenetics because histones influence what genes are in euchromatin and heterochromatin. By chemically manipulating histones, genes can be turned on or off by changing their affinity or availability to transcription enzymes. Most histone modifications are a process of methylation, acetylation, phosphorylation, ubiquitination, and summylation. It turns out that depending on the histone subunit modified and degree of modification, a modification can be a characteristic on both active or repressed genes. For example, acetylation is typically seen on active genes in euchromatin, while methylation can be seen on repressed genes or active genes.